Welcome, everyone. Thank you so much for joining us today. Encourage you to tell us what you do and where you work in the chat. And AGRO is a community, and we want to encourage that. And you see the QR code for information on AGRO division there. And we thanks to Eurofins Agroscience Services for their sponsorship, which is greatly appreciated for our Lunch and Learn webinar series. I'm your moderator. I'm the Vice Chair, American Chemical Society Agro Division, and I am and will be your program chair for the Agro Chemicals Division in 2026. We have a great community of scientists of advancing many areas of research in agrochemical science, and great program for the community, which includes things like this webinar series and a robust award program. And encourage you to organize a symposium and serve on committees. We're right at starting our proposing ideas and calls for papers for the 2026 meeting already. These awards, the fall meeting is in Chicago in late August of next year. Okay, so I just noticed that the look of Zoom has changed a little bit, but I guess the important thing to note is to see all the options. Use your cursor from your mouse to hover over the screen, and that menu will appear to say, for example, go into the Q&A or comment in the chat. Encourage everyone to be professional, courteous, and respectful, and please follow these common sense rules. Okay. Our first speaker, Dr. Thomas Selby, 45 years with DuPont, FMC, co-inventor of 4 agrochemicals. Sorry, I wrote that twice. BS University Penn, PhD, Indiana University, and our winner of the AGRO International Award for Research in Agrochemicals. And Tom and all his work and development of agrochemicals, holder of numerous patents. And with that, Tom, can you take over?
Yes, let me share my slide deck here. All right, very good. I'd like to just say that I was deeply honored to receive the award this year. It was definitely an exciting career capping milestone for me. And generally, I tell people that although I was trained as a synthetic organic chemist many years ago, actually, in graduate school, I've always had a strong interest in the biology behind programs. So the field of agrochemical discovery was always a natural fit for me career-wise. And even today, I still get excited when a new area of biological activity gets discovered. And if it's a new mode of action or if it has commercial potential, that's even more exciting yet. But the ability to do that has become more challenging over the decades. And our industry, like pharma, always tries to embrace new tools, methods, and techniques to discover these new areas. I've had the opportunity to work in all three ag disciplines, herbicides, fungicides, and insecticides, although I've always had the most passion for herbicide research where I began my career. Today's talk I thought would be a little different than the award address I gave last month at the ACS meeting. And since this is a webinar learning series, I thought it would be informative to talk a little bit about the process uh, involved in actually the discovery of new agrochemicals. And in my title here, you see the word art, and I put that in purposefully to emphasize that there is no guaranteed, assured way of obtaining success. There's always a certain amount of uncertainty and risk in this process. And frankly, a little luck along the way is always welcomed. I want to emphasize that discovery research definitely requires a teamwork. Not only do you have chemists and biologists making and testing compounds, but when a project gets going, very soon you have biochemists, enzymologists, molecular biologists trying to understand how the chemistry is working, what site of action might be involved. And also, at some point, formulation scientists get involved to find the best way of delivering bioavailability of the chemistry that's been discovered. And then finally, under the bracket of regulatory sciences, not only is toxicology very critical to know, but also early in a program, environmental fate, ecotox, understanding the physical properties. And when a program moves forward, a variety of activities can get triggered. And so it's very resource intensive. I've come to believe that there's uh, the most important part of this process is finding the right starting point in the very beginning, and the headwater, if you will, of the programs. And you want something that has a high chance of giving success at the end, rather than not leading to a potential commercial candidate. And I've come to believe that there's two main strategies. I think this is what we follow at FMC. Other chemists might look at it a little differently, but one strategy would be selection-based discovery. And that what is that? That's conventional and high volume screening of compounds from a variety of sources. And it can also include natural products. And so you're selecting for starting uh, that starting chemical compound. You're not designing it. And so you're sort of stuck with whatever mechanism of action you may find. But it is a successful way of doing it. Another way would be idea-based discovery, where you have a good understanding of the scientific literature, especially past and present patents. And you can even use computer-aided design tools today as well. But there you're picking your area of activity and a lot of times picking the mode of action that you want to get involved with. And so it's a little different approach, but both ways have been used successfully. I started my career working on a class of very potent sulfonylurea herbicides discovered by this fella, George Levitt, a great mentor to many of us starting our careers at that time. This is the general structure of sulfonylureas. And George was doing some interesting chemistry around reactions of sulfonyl isocyanates, and through a conventional screening operation I'll just mention, eventually arrived at probably one of the most potent classes of herbicides that have ever been discovered. And there was over a half a dozen products that were launched here by DuPont for just about every crop use. And that's very unusual at least then for one mode of action. And these compounds interfere with the biosynthesis of branch chain amino acids in plants by inhibition of this ALS enzyme. And this discovery, from my view, brought on an era of high potency herbicides where very little material is taken out into the environment versus conventional herbicides at that point in time. And you can see here this Glean container, small granular formulation with a small grad cylinder on top, just to give you an idea of how much of a reduction in size that was. And one of my first assignments was to work on the heterocyclic part of the sulfonylurea molecule. And this is a great way for me to jumpstart my career in ag discovery. And one of the most impressive parts of that program was to be able to see a compound go from greenhouse to field activity. And this is just one compound which became classic herbicide. It's a soybean compound, rather high use rate for a sulfonylurea. But you can see the untreated plot here where the weeds have totally overgrown the soybean canopy underneath, whereas this compound can take out selectively the weeds but leave the soybeans uninjured. And this compound is able to go glutathione conjugation selectively in soy. But this had such a dramatic effect on me, I think, it might explain my career-wise enthusiasm for herbicide research. We were always encouraged to go off to find new areas of biological activity because you wanted to keep this discovery machine going forward. And this is a class of bleaching herbicides I came across in the late 80s. This compound was picked up via conventional screening. And it turned plants white, albinism, which was very interesting symptoms and later shown to be an inhibitor of phytoene desaturase, a key enzyme involved in the biosynthesis of carotenoids, which protect chlorophyll from undergoing photooxidative damage by sunlight. And over several years, a wide variety of analogs were prepared here. Some very nice field activity, particularly in cereals. But we began to notice very long soil half-lives. And that became a significant issue where eventually we needed to drop the program because of the half-lives were just too long. And this was a valuable learning lesson for me earlier in my career, and that is you cannot just be focused on the chemistry and the biological activity. You really got to get to understanding what the environmental fate might be early on as well, and also some idea of what the toxicology might be. Interestingly, though, our work on these PDS herbicides led to a totally new area of fungicide research for us, which in turn gave rise to a commercial powdery mildew agent. And this pyridopyrimidinone had been prepared as a intermediate in that herbicide project, and it showed some interesting fungal activity, particularly control of powdery mildew. Obviously, it's important to test all of your intermediates in general screening because it might be a starting point for your next program. And here we made a wide variety of pyridopyrimidinones that I don't have shown. They had excellent, some of them had excellent greenhouse activity, but it was very disappointing translation to field activity. Fortunately, one of the things we did here was a core modification where this is replaced by what is called a quinazolinone. The nitrogen and carbon are swapped and the double bonds moved around. And Quinazolinones maintained excellent levels of activity, not only in the greenhouse, but in the field, which was a great breakthrough. And this iodo derivative was eventually selected for commercial development as proquinazide. And here you see a very clean barley field being evaluated. And this area of chemistry is not toxic to mildew spores. When an untreated one lands on a leaf surface, it generally grows a germ tube and then develops an aprosorium, which actually penetrates the leaf surface. Whereas a treated spore will grow that germ tube, but it never gets the biochemical signal to form the aprosorium. And so essentially, the pathogen is locked outside of the plant. I think that's a pretty cool mechanism of action. And to top it off, we were actually able to move from a fungicide area that involved proquinazide to a totally new class of rice insecticides that were discovered over a decade later. And how did that happen? This pyridopyrimidinone mesoionic compound, pretty yellow solid, was made as a side product in the proquinazide effort, did not have antifungal activity, sat in a file for 10 years, and then was picked up in a retro screening effort to have weak but interesting insecticidal activity. And this team of chemists continued to do optimization work here over several years. And eventually, this compound was selected for commercial development as triflumesoparam, a totally unique nicotinic agonist that is an excellent control agent for brown plant hopper, especially, which is a very serious plant pest in rice cultures in Indonesia. So, from a art of discovery standpoint, you see, we went from a herbicide intermediate to a fungicide program and product, and then from a fungicide side product to a totally new class of insecticides over a decade later. So just to point out that continuum. I do have one example of a natural product giving rise to a big class of very commercially successful fungicides. The first product to be commercialized here was a blockbuster broad spectrum fungicide by Bayer, azoxystrobin. And the starting point was this natural product, strobilurin A, a fungal metabolite, unstable, uh, and has an interesting methoxyacrylate toxophore on it. You can see in this commercial product that same methoxyacrylate piece serving as the toxophore, although the rest of the molecule is different. And quite a few products came out of this area, a fairly big class, very successful. And these are complex 3 binders to involve to the component of the electron transport chain process in mitochondria. And it's a specific site on the complex 3. And Corteva has been very successful with pursuing natural products as a starting point. And they have a new class of fungicides that also interfere with complex 3 on the electron transport chain, but they bind to a different site there. And that is very exciting. And so, again, this type of approach can be successful in finding new agrochemical products. One of the most exciting programs that I was involved with in my career were the diamide insecticides, IRAC group number 28, 2 members here, Rynaxypyr and Cyazypyr. And Rynaxypyr is a tremendous control agent of chewing insects, including lepidopteran pests such as tobacco budworm, fall armyworm, etc., coleopteran pest as well, beetles, whereas thiazapir has a lower log P, higher water solubility, and has an extended spectrum of activity, going not only good activity on these two insect types, but improved activity on what we call sucking piercing insects, insects that feed off the fluids of plants, represented here by these hemipteran pest aphids and especially whitefly. You can see here not a big change going from chlorothiocyanate, but I want to emphasize that a lot of compounds were made in this interim looking for compounds with the right uh, level of cross-spectrum efficacy. And these compounds were shown to be modulators of the ryanodine receptor. They bind to an allosteric site on the ryanodine receptor separate from that of the natural product and cause muscle cell contraction and muscle paralysis in insects. And a very interesting discovery story here, how the SAR evolved. And this would be more of an idea-based strategy where my colleague and friend of many years, George Lam, was following up this phthalic acid diamide that appeared in the patent literature application, unknown mode of action. And George was interested in getting some novel IP, intellectual property in the area. And he switched this amide around so that now instead of a phthalic diamide, you have an anthranilic diamide. And when he did that one change, he lost the activity. However, when he did another change at the same time and take this methyl group and rotate it down here, he obtained this compound where most of the activity came back. And this was a very exciting in-house lead for us. And lots of compounds made over multiple years, actually thousands of compounds eventually made. And I'm just going to say that I became interested early on in this program with looking at heterocycles here, particularly pyrazole. And this particular compound, where you have a chlorophenyl attached to the pyrazole, was prepared by my colleague John Freudenberger, and it had commercial levels of activity. This was really exciting to us. We knew we had something here that had a chance of making it all the way. And then my other friend and colleague, Tom Stevenson, who got involved with this area, switched out the phenyl by a pyridine and eventually introduced chlorine at this position here. And this particular compound that was first prepared in Tom Stevenson's laboratory became Rynaxypyr. Unprecedented levels of activity, just a tremendous product. I think it's selling close to 2 billion annually in sales, just phenomenal. And I have to say, we could not have asked for a better program to come along at that point in time than this one. So it was a lot of fun and very exciting. This is what we sometimes refer to as the famous cabbage photo. This shows Rynaxypyr controlling at 5 grams per hectare both diamondback moth and cabbage looper, whereas the cabbage treated is nicely protected versus the untreated rows shown here. And this was the first synthesis of thiazapyr, was prepared in my laboratory by my associate Ken Hughes. We had some difficulty initially in making this compound, but eventually got there and eventually came up with better ways of making it. The discovery stories have been published if you're interested. And this compound, as you can see, has that extended spectrum against a hemipteran pest such as whitefly, the cabbage here being nicely protected versus the untreated nicotinic check. And here are two advertisements. This is a foliar applied formulation under the trade name AGRO for thiazapyr active. And this formulation is for soil applied applications, Verimark, again powered by SAI, is Superior Active. And there's nothing more rewarding to me, I feel, more validating to actually see a compound from an area that you were involved with go on and get is in a commercial container. I think it's validated for what we do in the laboratory, and quite exciting to see. I just have one slide I want to show very quickly of a tremendous omycete curative fungicide that was discovered by my friend and colleague of many years, Bob Pasteris. He followed up on this compound that came in a library of compounds that were evaluated in high throughput screening. And it had such weak activity, I would not have probably have followed up on it, but Bob did. And a team of chemists worked here and scientists. And eventually they give just a spectacular compound. You could see here doing a very nice job controlling potato late blight, totally new mode of action. And there's a paper on this discovery as well. This paper actually appeared on the cover of that journal issue. So quite an impressive find here. And then finally, I just want to talk about the discovery of tetflupyrolimet, which is a totally new mode of action herbicide that interferes with pyrimidine biosynthesis in plants and deprives the plant of a very critical nucleotide building component. And this is the first totally new mode of action that's been uncovered in over 30 years. And as a result, it gets a new IRAC group designation, a number 2028. You can see the structure of the molecule. This SS stereochemistry is very important for activity, and it has the trade name Dodalex active. I wanted to show this very scenic photo of a rice paddy to emphasize our strong interest in this compound for weed control, particularly grass weed control and rice production worldwide. And I should also point out Atul Puri, who was the lead biologist, that was not only played a key role in the discovery, but also in the development of this compound, and right now the commercial launch, which is going on as we speak. So very exciting time. So how did we find this compound? Through a selection-based approach, high throughput screening. It's not a high success operation probability-wise, but every now and then interesting things do come through. And this is one compound that caught my eye, this amide substituted pyrrolidinone. And my friend and colleague of many years, the late biologist Mark Ruggiero, pointed out to me that it interfered with the growth of grasses as if they were not getting a nutrient required for growth. And that was very interesting. In our counter screens, we're able to roll out existing or known modes of action. But one of the things we do right off the bat with a compound being tested from the outside is to verify its structure. And the mass spec was pretty good, showed the right molecular weight. The NMR looked good, the right number of protons, except one peak was problematic to me. So I actually prepared a sample of the compound, and the NMR did not match and was inactive. So that was a uh-oh moment. But on thinking about it, with the NMR being so close, thought maybe this amide group is over here at the 3 position. And so I prepared that compound, and the NMR did match and had the right peak here and had biological activity. So we were off and running to make a variety initially of racemic analogs. Then we're able to make enantiomer rich derivatives because stereochemistry is very important in this area for activity. This particular compound that has an N-methyl group here became tetflupyrolimet. I just want to point out here, you see 2 aggressive growing grasses, late water grass, barnyard grass. they're being nicely controlled at 125 grams per hectare. But if you focus in on this pot, you see very nice safety to not only transplanted japonica rice, which is a big practice in Japan, but you also see very nice safety to direct seeded indica rice, which is very popular in places in Asia as well. And you think about it, it's very challenging to be able to control a monocot or grass weed in a monocot or grass crop such as rice. But this area has the ability to do that, which is quite amazing. And I did mention the compound interfering with pyrimidine biosynthesis in plants, and it was actually shown to interfere with this particular enzyme, dihydroorotate dehydrogenase, which sets on the mitochondrial membrane as part of the machinery that cranks out DNA and RNA. And without this nucleotide building component to be incorporated into particularly RNA, there's a detrimental effect to the plant. And it's believed that this chemistry binds to the ubiquinone binding site on dihydroorotate dehydrogenase. I just want to mention that the team of scientists at FMC did a fantastic job in detective work and using some of the modern tools and techniques that are available now that were not available in the beginning part of my career to figure out very quickly how different types of chemistry are working at the biochemical level. And they were actually able to isolate the gene that codes for this enzyme. A very interesting story. And they have a publication in this prestigious journal, the Proceedings of the National Academy of Sciences, if you want to read more into how that was done. Just some incredible work there. And there was multiple years of successful field testing of Dodalex active. You can see here transplanted indica rice, treated versus untreated, and a nice plot from South America with the trade name Canali shown there. The discovery story was published in the Journal of Ag and Food Chemistry, and it actually appeared on the cover of that issue as well. And I want to emphasize that there's a lot of teamwork that goes into the discovery process. This was the tetflupyrolimet team of folks, but actually, when it goes forward, you have teams of people working in regulatory science on registration, process chemistry, plant scale up, field testing worldwide, et cetera. So it's a really costly and resource intensive operation. And I want to emphasize teamwork there is very critical. I did retire last year and I like to fly fish. Those of you who know me, this was a fly-in trip to Alaska. And this is a famous park known for its brown bears, many brown bears with photographers, and they're roaming all over the place, well fed on sockeye salmon. So uh, that's a good thing, although you keep one eye on them. And I always tell people discovery chemists and fishing has a lot of similarity. You have tools, techniques, but you have no guarantee for success, and you definitely don't know what you're going to get at the end. And I think both of those uncertainties with both activities is what makes it so interesting to me. But I'll end there, and thank you for listening to me. And again, I'm extremely grateful to have received the honor at this year. Thank you very much.
Thank you so much, Tom. That was a fascinating presentation.
Oh, thank you, Dina.
Okay, our next presenter, Dr. William Mitch, Professor, Department of Civil Engineering and Environmental Engineering at Stanford University. His MS and PhD from University of California, Berkeley, BA from Harvard, Winner of the 2025 Journal of Agricultural and Food Chemistry Paper of the Year. His work focused on environmental chemistry with applications in municipal drinking water treatment, wastewater treatment, and recycling, and in agriculture. And so take it away, Bill.
So first, like Tom said, thank you for the award, and I'll be talking about the subject matter of that today, which will be a little bit different from the previous presentation in that I'm in civil and environmental engineering, so we tend to focus on things like pollution control, and so we're trying to develop a technology to capture and degrade sulfuryl fluoride fumigant. So what is sulfuryl fluoride? Most of you probably know, but just to kind of recover this, it's a fumigant used to kill insects. It's the main component of the fumigant used to kill termites in structures at about 10 grams per cubic meter for a 2-day contact time, and the contact times will be important a little bit later. It's also used to control pests in durable agricultural products prior to export. So you have these fumigation chambers. Let's see if I can get my little pointer here, such as the following, where you put in nuts or other durable agricultural products, expose it to sulfuryl fluoride gas at higher concentrations, but similar contact times, 1 to 2 days. And to kill the pests before you export things like almonds to China, for example. It's become very important, particularly since methyl bromide, which was the previous gas used for these purposes, was phased out over concerns that it was an ozone depleter. The challenge is that in both of these applications, the sulfuryl fluoride is vented to the atmosphere after use. And so the atmospheric concentrations have been slowly increasing after the phase out of methyl bromide. And one of the challenges is that as a gas, it has a 5,000 times greater global warming potential than CO2. And so there are concerns, particularly for the agricultural exporters, as to whether places like the EU might start to restrict imports of U.S. agricultural products over concerns about these greenhouse gas emissions. So we are, oops, hold on a second here, operate the controller. So what to do? One option is to find an alternative to sulfuryl fluoride fumigant. They don't currently exist. There are other fumigants, but the contact times tend to be longer than 1 to 2 days, and those don't seem feasible, particularly for applications such as moving crops fast out of ports. So another alternative is to say use the gas, but instead of venting it directly to the atmosphere, try to capture and destroy it after its use. So one option is to take the, here, get my pointer back. After you use the fumes in a fumigation chamber, run it through a countercurrent aqueous scrubber system where the water contains a reagent that can degrade the sulfuryl fluoride gas. So early options used hydroxide, so high pH solutions, which can react with sulfuryl fluoride as a nucleophile and knock off the fluoride. The challenge is that it works, and you can capture and degrade about 90% of the sulfuryl fluoride fumes, but you end up making this fluorosulfite anion, and the toxicity of that intermediate is unclear. So one of our collaborators at the Connecticut Ag Station, Joe Pignatello, figured out that hydrogen peroxide, its conjugate base, so hydroperoxide anion, is also a nucleophile, but much more potent. And so if you can get access to this conjugate base of hydrogen peroxide, you can completely defluorinate the sulfuryl fluoride and form sulfate and fluoride as relatively harmless salts. It works within about 15 minutes contact time to get the complete defluorination. And you also need kind of tight pH control right near the pKa of the hydrogen peroxide. Because if you have higher pH, you have obviously more hydroxide, and then you'll start building up this fluorosulfate anion. So there's our collaborator Spencer Waltz, who works at the USDA, has been doing pilot tests of the scrubber technology at fumigation facilities at places like ports. So this is for the agricultural fumigation sort of application. So the higher concentrations for 1 to 2 days in these 76 cubic meter containers. This translates into about 10 kilograms of sulfuryl fluoride, or about 100 moles of sulfuryl fluoride, that are vented from the fumigation chamber through the countercurrent scrubber shown here over about 2 hours. Again, the time scales will be important later. And we can vent all these fumes, about 98% venting of the chamber within about 2 hours, run it through this countercurrent scrubber. The scrubber solution is concocted from these high concentration stocks of potassium hydroxide and hydrogen peroxide. So we buy these barrels, mix up a batch at the port, run it through the scrubber, and they've been making up batches that are about 5 times more moles of hydrogen peroxide relative to what you need for sulfuryl fluoride scrubbing. So you can scrub about 5 fumigations worth of sulfuryl fluoride, but a couple of challenges with that. One, it's expensive to buy these reagents. You got to ship them, and you got to deal with the CO2 emissions of shipping these reagents. And probably most importantly, it's hazardous to the workers on these scrubbing systems, because you're handling these high concentration stock solutions. And when you mix the potassium hydroxide and the hydrogen peroxide, it gives off a lot of heat. And in fact, in one of the pilot scale tests, the scrubber melted. So, some of the other challenges faced by this kind of ongoing pilot tests at these port facilities is that as you scrub with these high concentrations of hydrogen peroxide, with each fumigation, you're degrading or using up the hydrogen peroxide, and you're making fluoride and sulfate at about the 2-to-1 molar ratio you'd expect from this nucleophilic substitution reaction. And so you're accumulating all these salts. What do you do at the end of this? After 5 cycles, you've built up substantial levels of waste salts. And how do you dispose of this? Particularly for things like fluoride. So it's obviously less toxic than the sulfuryl fluoride, but it's still challenging to get rid of fluoride. So one of the things they've been testing out at these scrubbing pilot tests is to precipitate the fluoride with calcium, essentially make calcium fluoride, and maybe you can sell this as a commercial product. So they add in various sources of calcium, precipitate the calcium fluoride, and press it into a cake using one of these belt or filter type presses. What we've been looking at was an electrochemical alternative to generating the reagents needed for the scrubbing operation, the hydrogen peroxide and the hydroxide on site. So can we just make the reagents there instead of buying these barrels of high concentration stocks and mixing them on site and giving off all this heat from that mixing, and avoid these kinds of dangerous scenarios, while ideally also saving money? And so the goal essentially is to replenish the reagent we needed for the scrubbing solution on site between the fumigation cycles. So again, the timing becomes important because we have about a day's worth of fumigation, and it only takes 2 hours to vent and scrub the material. So in between, say on the order of 20 hours, can we replenish our reagents in time for the next scrubbing event? So the benefit of this would be that we'd avoid buying and shipping these high concentration stocks and the danger of mixing these reagents on site. One of the other things I'd point out is that hydrogen peroxide degrades itself. So it'll react with itself, make through a disproportionation reaction, oxygen and water. So if we only need about 200 millimolar hydrogen peroxide per scrubbing event. So if we can make that in between each fumigation cycle, then we can essentially make it at a lower concentration versus the 5 fumigations worth that they're currently making at the site. And so since we have a lower concentration of starting material, we'll minimize the losses of hydrogen peroxide through this disproportionation reaction. So again, the scheme would be you fumigate your durable agricultural products, run it through a countercurrent scrubber. Sulfuryl fluoride wouldn't go out to the atmosphere. But during the scrubbing event with the conjugate base of hydrogen peroxide, you would take the sulfuryl fluoride and convert it to fluoride and sulfate. Now you have this high salt waste product, but if we run it into the electrochemical system, maybe we can regenerate our hydrogen peroxide and hydroxide for the next scrubbing event. So what we've been using is this 3-chamber electrochemical cell shown down here in the lower right, where the active portion is the cathode, where we're taking oxygen and reducing it to hydrogen peroxide and hydroxide, so the 2 reagents we need simultaneously. And we're doing that through a gas diffusion electrode. So we have oxygen gas that comes in this chamber over here on the left. We have a permeable cathode. The oxygen travels through it. And so you get higher concentrations of oxygen at the active surface of the cathode. That's important because oxygen is poorly soluble. So we're boosting the oxygen concentration at the cathode surface. The anode just completes the circuit. We're oxidizing water to oxygen gas. It also generates acid. So we want high pH in the catholyte and the hydrogen peroxide. So we put this cation exchange membrane in between to keep the 2 chambers separate. If we didn't have this, the acid in the anode would just neutralize the base in the cathode, plus the hydrogen peroxide would travel over to the anode and get oxidized back. So we would lose our product. So it's important to have this 3-chamber system. So first, does it work? And the answer is yes. So what we're showing down here is time and the accumulation for different conditions of salts relevant to a used scrubbing solution, either concocted in the lab or an actual scrubbing water from one of these pilot scale operations. And we're showing the accumulation of hydrogen peroxide over time. So we're generating on the order of 200 millimolar hydrogen peroxide, about the concentration we need for the next scrub, within 4 hours, and we have on the order of 24 hours to do it. So we're well within our time frame. Now, what I want to do is take a little aside because there's a lot of interest in my field, environmental engineering and waste control, on electrochemical systems because of this promise of making reagents on site, tunability. You can just plug the device into the wall and use electrons, that sort of thing. But it rarely works out. It's always kind of restricted to lab scale systems, and wanted to kind of contrast perhaps why this particular application is an excellent option for scaling up, why it's different from some of the failures we see in our field in the past. And part of that is that our typical research applications in environmental engineering are pretty challenging. Most people are looking at oxidizing trace contaminants, micrograms per liter of, say, pesticides or something of that nature, in matrices such as drinking water or other high flow rate streams like municipal wastewater. Sulfuryl fluoride scrubbing sort of addresses some of the main challenges faced by what's typically goes on in our field. So, wanted to kind of go through those challenges. Challenge number 1. Electrochemistry needs high salt concentrations to make the system efficient. You need high conductivity to lower the solution resistance and therefore lower the voltage. How do you get those in a typical application? You've got to buy the salts, and you need substantial levels of salts, 0.1 molar or higher, to keep that conductivity low. In many of these applications we're looking at in environmental engineering, you're dealing with low salt concentrations, drinking water or municipal wastewater, so you need to buy the salts. And it's not the electricity that costs so much, it's the cost of the salt to generate the conductivity that tends to dominate the operational costs of those systems. Here with sulfuryl fluoride, we're generating about 0.1 molar sulfate and fluoride with each scrubbing application. You can see that here. And so we're getting free salts as a waste product of the previous scrubbing operation. And so what we're showing here is the cell voltage as a function of numbers of scrubs, or the equivalent salt concentrations that would be generated from repeated use of this scrubbing solution as these salts accumulate. And what we see is the cell voltage declines with increasing reuses of the solution, and the energy needed to generate a cubic meter of the scrubbing solution declines accordingly. The other thing is to think about is in many of these lab scale applications, say for drinking water applications, you can kill the micrograms per liter, let's say, of contaminant, but by adding 0.1 molar salts, you've now converted the fresh water into salt water. Nobody wants to drink it anymore. But with the sulfuryl fluoride waste solution, we're actually in an ideal situation because that waste solution was a disposal problem. How do we get We talked before about how do you precipitate fluoride as calcium fluoride, for example, to make it safer for disposing of these waste salt solutions. Now we're taking this waste salt solution and valorizing it, turning it into a useful product because it's a free source of conductivity for our electrochemical system. Challenge number 2. Electrochemical applications in the drinking water field tend to focus on oxidizing contaminants at an anode. But real water always contains things like chloride and bromide, and the anode tends to oxidize these to toxic products such as chlorate, perchlorate, bromate, that sort of thing, and substantial concentrations, in some cases milligrams per liter. We're avoiding that because for the sulfuryl fluoride scrubbing application, the active reagent is produced in the cathode on the reducing side of the system. And even though we will have salts that are in the anode chamber, sulfate and fluoride, for example, these salts aren't readily oxidizable to toxic products. They tend to be inactive, unlike things like chloride. Expensive electrode materials. If you're looking at focusing on anodic systems, so oxidizing contaminants, anodes tend to be made out of very expensive materials. Boron, doped diamond, those sorts of things that can withstand these harsh acidic and oxidative conditions. For sulfuryl fluoride, we're focusing again on the reduction at the cathode side of things. And cathodes can deal with inexpensive materials like carbon. So that's what we're using is carbon paper coated with a carbon black catalyst. They're stable under reducing conditions. They would not be stable in the anode, but we're focused on the cathode. A note on the carbon black, what is it doing? When we're reducing oxygen, we have two choices. We can feed in 2 electrons and make what we want, hydrogen peroxide and hydroxide, or we can feed in 4 electrons and just make hydroxide. Well, the carbon black makes it selective for this 2 electron reduction over the 4 electron. So here we're showing hydroxide accumulation over time with and without the carbon black, and that's comparable. But when we look at the hydrogen peroxide, we get substantially more hydrogen peroxide with the carbon black. Challenge 4, low efficiencies. Oxidizing micrograms of contaminants in a real drinking water matrix where you have milligrams per liter of natural organic matter. The electrons are going to go are going to focus on this much higher concentration constituent than the contaminants of choice. So the coulombic or Faradaic efficiencies, the fraction of electrons that goes towards useful reactions, are typically very low, less than 1%. But with sulfuryl fluoride, we're focusing on reducing oxygen to hydrogen peroxide. This is present at milligrams per liter. And so in some of our experiments, we're seeing Faradaic efficiencies up to 99% in some cases. So much more efficient. And lastly, slow kinetics. So when we're oxidizing micrograms per liter of contaminants in something like a drinking water, we need for something to be viable for a water treatment process to have a treatment plant with a reasonable footprint. We need to treat that water in relatively short amounts of time, about 15 minutes. But many of the lab experiments involve taking a batch of water and frying it for something like 24 hours. That's not necessarily going to work. With the sulfuryl fluoride fumigation system, we have ideal timescales. So like we mentioned, the fumigation takes place over 1 to 2 days, We then vent the fumes through the scrubber over the course of about 2 hours. And what we need to do in the interim, when we're fumigating the next batch, is to regenerate our reagents. So we have on the order of 1 to 2 days to regenerate for the next batch. And we already showed you this plot where we could get the sort of 200 millimolar of hydrogen peroxide within about 4 hours. So just show you a couple of quick results here. We've been looking at various performance metrics, things like the Coulombic efficiency, again, the fraction of electrons that go towards useful products like hydrogen peroxide, the time to get to the amount of hydrogen peroxide we need at the appropriate pH, the full cell voltage, so that's going to translate into power, shown here, the energy required to produce a cubic meter of scrubbing solution at the desired hydrogen peroxide concentration. Oops. So we've been looking at different effects, such as, for example, electrical conductivity or the salt concentration. So operating at a particular current density, as we increase the salt concentrations to levels reflecting the salts produced after half of a fumigation scrubbing, 2 scrubs or 5 scrubs, the salts increase as they accumulate in the waste scrubbing solution. And what we see is that as the salts increase, as the associated with the scrubs down here, as we saw this before, the full cell voltage declines, and so does the power required to generate a cubic meter of scrubbing solution. The time needed to get to 250 millimolars remains 2 to 5 hours, so well within that 24-hour period. And up top is shown the efficiency, so all over about 50%, so pretty efficient. Another example here at relatively low salts, but increasing the current density, so essentially pumping current in faster to increase the kinetics of the reaction. And indeed, what we see is to get to 250 millimolar hydrogen peroxide, the time scale goes from about 2.5 down to 1 hour. We lose some in terms of Coulombic efficiency, because as we're pumping in electrons, there's going to be side reactions that start hogging up some of the electrons, but still pretty high current efficiencies. So as the current increases, the voltage goes up mildly, as does the energy required to generate the hydrogen peroxide. So it starts to get less efficient, but we're already at only 2.5 hours, even at the lowest current density. And lastly, we've been looking at increasing the catholyte volume to cathode surface area. So essentially, with the same electrode surface area, can we treat more water? That will ultimately translate into cutting down the capital cost associated with the electrodes. So what we're showing here, as the catholyte to cathode surface ratio increases, the time required to get to our target reagent concentration increases from, say, 2 to 9 hours. Still well within our 24-hour period. It increases because with the same electrode, we're treating a lot more water, so it's going to take a lot longer to ensure that all that water reaches that 250 millimolar concentration. But we're still at high current efficiencies and relatively low voltages. Lastly, we kind of did a little quick cost estimate here for a pilot scale scrubbing water at the conditions shown here, comparable to what we saw before. Again, we saw this plot before, but if we take the numbers of the power, voltage, etc., associated with this run right here, and we use that to calculate the cost for operating costs, at least, for running this system. So here's the current mode of action, purchasing the reagents and mixing them up on site to run the scrubber. We're running at about $18 per cubic meter of scrubbing solution to buy the sodium hydroxide and hydrogen peroxide. If we run it, if we generate the reagents electrochemically, the main cost is the electricity, Before I said it was the salts, but we're getting free salts here since we're using the waste scrubbing solution. And so all we have to pay for now for operating costs is the electricity. And it depends on electricity is more expensive in California than in Texas. But what we can see is we're still, even in California, below the cost of purchasing these reagents. So just to kind of quickly summarize, the electrochemical benefits here are lowering the operational costs. We're avoiding the purchase and transport of the chemicals and avoiding the associated carbon dioxide emissions. A big one, I think, is safety, particularly if we want to apply this for something like home fumigation units, the termite applications, for example. Having local contractors run these systems, the safety aspect is going to be very important. So avoiding mixing these high concentrations of chemicals on site and the associated heat emitted from these systems. Instead, we can produce it on site under more controlled conditions. And lastly, we can ideally get a more efficient use of the hydrogen peroxide. So I mentioned that if you mix up batches of very high concentrations of hydrogen peroxide, you'll start to lose some through this disproportionation reaction. And that's what's shown here, where we're looking at a difference as the hydrogen peroxide is used up. We're looking at how much went towards useful reactions versus were wasted by disproportionation. And there was a gap there showing that some of this hydrogen peroxide degrades. What we're doing right now is working on some other aspects. One is addressing the capital cost. I had mentioned that we're focusing on the cathode, which can involve inexpensive materials like the carbon that I mentioned. But it is true that at the end of the day, we have to have an anode to run the system. We're getting some benefits in that we can control what's in this analyte. But in particular, one of the things we're challenged with is that fluoride tends to etch the catalyst in the anode. And so here's an anode made out of iridium oxide, and you can see hopefully that there's some discoloration here. It's essentially indicating that the iridium is eroding away from exposure to hydrofluoric acid under the acidic conditions of the analyte. Iridium's very expensive. So what we're trying to do is work with our USDA partners on ways to optimize the precipitation of the fluoride as calcium fluoride to minimize the residual fluoride in the analyte. We're also looking, do we need to use iridium? Iridium's very expensive. Can we use alternative materials that are cheaper and bring down that capital cost and test how long they last? We're also looking at scaling up the system to enable a full operating and capital cost estimation to try to get towards this type of application that we saw before in ports. Right now, our lab scale anode or electrochemical system is pretty small. We're scaling it up tenfold. Still doesn't look that big, but this is 100 square centimeter electrode. That's about as large as you can feasibly get and still distribute the current efficiently. So for an application like this, what we would do is use a lot of these as modular units and essentially stack them. So lastly, I'd like to thank USDA for the funding and some of our collaborators, and move on to the Q&A session.
Thank you so much, Bill. Very creative, innovative, and amazing, just all the work that you've gone through to mitigate the environmental impacts in a practical, economical way. Thank you.
Thank you.
Yeah, Dana, you think we have time for about 3 questions?
Absolutely. Let's go for it.
Yeah. One is pretty specific to Bill, and I'll lead off with that, and then we can bring in Tom on camera after that. From Previn Dubey, Bill, as sulfuryl fluoride is known to hydrolyze in the presence of water, what would be your recommendation for fumigation when samples contain that higher moisture content? And then would increasing the concentration and extending the exposure time of sulfuryl fluoride help ensure that enough gas remains available to achieve effective fumigation?
Well, thanks for the question. I should just note that my expertise is not necessarily on the fumigation side of things. However, if we the hydrolysis with water itself, not with hydroxides. If you're not dealing with high concentrations of hydroxide, should be relatively slow. So, uh, the flip side of this is the fumigation contact times for the applications we were talking about are 1 to 2 days. So you probably will be losing some in a more humid atmosphere. But I don't, off the top of my head, since we don't deal with the actual fumigation side of things, have great answers for you on that front.
Great. Uh, Another question, and I'm going to merge about three of them here. And Tom, if you're still on with us,
Yes.
that's awesome. Uh, and this applies to you, Bill, too, and we can bring you into this question too. We have the discovery of new compounds undergoing a process with certain endpoints and certain outcomes that are favorable to move these things along, uh, such as uh, uh, such as things like systemic activities, physical properties, and uh, and human environmental toxicology. With the emergence of new technologies such as artificial intelligence, does it change the process? Does it change the metrics we're looking at? How do you think that's going to change in the future?
Did you want me to respond to that,
Yeah.
Tom here? Uh, yes. Throughout my career, particularly in the later part of it, I come to realize that getting the right balance of attributes for any research area, any compound area is extremely important. We chemists love to make compounds and you get activity. It's very encouraging, but you really do have to think about all of the attributes and balance, and particularly around not only environmental fate and toxicology, but the physical property part is extremely important as well. I think that's one of the most important parts of a discovery program is understanding the physical properties thoroughly. And that would hold for herbicides, fungicides, and insecticides because it's going to determine whether you have systemic activity, whether you have only contact activity. And, we had that in the diamide insecticide area where we had great activity. And initially, we had great activity on chewing insects, but we knew we had the potential to control insects that required more systemic properties, and so that was very important that we go in that direction. I mentioned early in my talk about the different strategies that we follow, and two main ones, but under idea generation strategies, which would be a design approach. I think computer-aided drug design is going to become more and more important as we go into the future, especially AI. And the question is exactly how AI is going to work itself into the design of new agrochemicals, where you might target certain pathways, certain enzyme targets, and how can you inhibit it? That has had um, that has had challenges to date because of one big issue, and that's translation from in vitro activity to whole organism activity. And it's not always a straight line. And trying to understand that better too is going to be very important going forward, is to take a great inhibitor on an enzyme and make sure it's able to work on the whole organism. So I think it's definitely a direction for the future on how AI is going to incorporate itself. It's also already making inroads to pharma and agrochemical research.
You have to bring Bill into this question.
Yeah.
How's AI going to affect things? And we did have a question about other technologies such as solar panels that might make energy availability to your processes more robust.
Yeah, so just about the AI question, I think it'll be pretty important in pretty much every field, but I think luckily there's still going to be room for humans, ideally. And so, in particular, I think AI can be useful for playing out scenarios such as, there's a subtle functional group modification. How will it behave in a treatment process or as a herbicide or something of that nature? But hopefully, where the human comes in, and I think Tom's talk showed this as well, is to think outside the box and say, where else could I apply this scheme? So, for example, it seemed like in Tom's presentation, he in several cases started with something that was an herbicide and then said, hey, maybe this molecule could also be used as a fungicide or something of that nature. And similarly, in my presentation, I focus on municipal wastewater treatment for the most part. Maybe there's an application where that makes sense where I could take this and apply this to this agricultural system here. And I don't see AI doing that at least anytime soon. I think it will help speed up our ability to accomplish the nuts and bolts of a project, but maybe the sort of the out of the box thinking is still in the human domain.
Right. I guess the last question, there was a lingering question about PFAS and forever chemicals, right? That's certainly an adjacent problem to yours, Mitch, Mitch, and Tom. You mentioned drug discovery and AI. What are some of the challenges that other fields tackle that might help inspire you to solve problems in pesticide discovery and environmental mitigation?
Well, fluoride is an interesting topic. I was actually going to ask you, Bill, about your process electrochemically around the question of HF from a safety standpoint. Do you feel for operators, is there any chance that HF, I think you showed that HF can be generated in the anode, and I was just curious, do you have any concerns around operator safety from, etching or, the fluoride from a mammalian safety standpoint?
Uh, no, not so much on that front. So the pH of the analyte gets down to about 2. That doesn't mean on a localized level, right at the anode interface, we probably get even lower pH in sort of the stagnant film layer right next to the anode, where we might be generating some HF and hence the etching. Like I mentioned, what we're currently trying to do is to optimize the precipitation process to minimize the fluoride in the solution. But I think our focus mostly now is on preserving the anode for as long as possible to minimize the overall operational cost. But I don't see it necessarily translating into a health and safety issue, for example, with like HF fumes rippling off of the analyte or anything of that nature.
Yeah, that was just popped into my head. To get back to you, Solito, on fluorine and agrochemicals, that's a hot issue these days, particularly with registering a molecule, for example, in Europe. They're very sensitive to that. And so there's a big push in in the agrochemical community to minimize or move away from certain types of fluorine containing or substitution patterns on molecules. It would be an advantage to be able to do that. But there has always been something magical about fluorine and particularly CF3 groups in really great working agrochemicals. So that is a topic where there's a lot of discussion on what alternatives in the future might there be, so that you have, environmentally, you have a clean bill of health. Yeah, definitely.
I'll just mention with the same electrochemical scheme, we have a different scheme for hydrophobic organic contaminants, some of the classics like PCBs and polybrominated diphenyl ethers. We're ramping up the difficulty scale where if we take activated carbon, which is the go-to treatment for hydrophobic chemicals, you absorb the material, but you don't kill it. But if we then feed electrons through the carbon, we can dehalogenate them, which is sort of the source of the toxicity, carbon chlorine, carbon bromine, etc. We're also starting to look at things like pesticides that have the occasional CF3 or CF bond, and we believe we will be able to defluorinate those. But when you mentioned at the beginning PFAS, that's a bridge too far. You need really extremely low voltages below what water can handle to achieve that. If you try to do it in an aqueous system, you just start generating hydrogen gas by reducing water, essentially. So, but I think for some of these kind of occasional fluorine groups on larger organic molecules, it's achievable to capture and degrade those chemicals with just a glorified electrochemical activated carbon column.
Great perspectives. Well, Dana, what a great session today. You want to close us out?
Oh yeah, thank you both. Yeah, it's been amazing. Well, coming up next Wednesday, we have two more award-winning speakers. Technology and chemistry shaping the sustainable future, and Tejas Shah, one of CNE News' 2025 Talented 12, a great honor, and also Spencer Walls is coming, and he won the award for innovation in the chemistry of agriculture, and you've already seen his work alluded to today. So, and then more webinars upcoming in October, November, as you see listed here. Again, thank you everyone for participating, and make one final pitch to join AGRO and division of ACS, and I'll participate in all our great work that we do. And you can even if you're not an ACS member, you can join the division as associate, and for I think it's $16 a year. It might be a discount for the first year too. So anyway, that's it for now. Thanks again for everyone, and have a great day.
Thank you.
Thank you.